We're encouraged by the early data we've seen in this phase 1 trial. We believe the next generation ADP-A2M4CD8 product is active. As of the data cutoff for the SITC presentation, we had 2 confirmed RECIST responses. One at 1 billion cells, which is a lower dose from those required to achieve responses with the first generation product. Also, we saw initial tumor reductions in 5 out of 6 of the first patients treated. These data support continued investigation of the next generation SPEAR T cells, focusing enrollment for the Surpass trial on patients with gastroesophageal, head and neck, lung, and bladder cancers. We will also initiate a phase 2 trial with this next generation product for patients with gastroesophageal cancers next year. I'd like to briefly describe the design of this trial and provide a little background on this next generation product. The Surpass trial is the first clinical trial with our next generation SPEAR T cells targeting MAGE-A4, known as ADP-A2M4CD8, that co-expresses CD8 alpha along with the engineered TCR directed against MAGE-A4. This trial is designed to evaluate safety and efficacy. The figure on the right illustrates that co-expression of CD8 is intended to achieve greater cytotoxic function of CD4 positive or helper T cells by enabling them to kill cancer targets while still maintaining their helper function. Preclinical data we presented at AACR last year support this, and we saw that CD4 cells targeting MAGE-A4 that also have CD8 not only killed MAGE-A4 target cells, but they also secreted more cytokines and chemokines, the so-called helper function of these cells, that potentially serves to recruit more immune cells into the tumor site and increase responses. The Surpass trial utilizes a modified 3+3 dose escalation design, but unlike our other phase 1 trials, there are only 2 dose escalation cohorts. In the first 2 cohorts, we treated patients with a predefined stagger between patients and dosing groups to evaluate safety. To be eligible, patients have to be HLA-A02 positive with advanced cancers that express the MAGE-A4 protein. T cells from patients are isolated, transduced with a lentiviral vector containing the MAGE-A4 TCR and CD8 alpha, and then expanded. Prior to SPEAR T cell infusion, patients receive a lymphodepletion regimen of cyclophosphamide and fludarabine. After SPEAR T cell infusion, patients are followed for safety and anti-tumor response using RECIST 1.1 criteria until disease progression. The data cutoff for this presentation was October 1st and includes 6 patients from the first 2 dose escalation cohorts who received doses of up to 1.2 billion and 6 billion transduced cells, respectively. There were patients with multiple tumor types, 2 with esophagogastric junction cancer, or EGJ, and 1 patient each with myxoid round cell liposarcoma, ovarian, head and neck, and esophageal cancers. Patients ranged in age from 31 to 71 years. Most patients had an ECOG score of 1, and all had received at least 3 prior lines of therapy. This table shows adverse events that occurred in more than 1 patient, with AEs of interest highlighted by the red boxes that had been captured as of the data cutoff. The most common adverse events were leukopenia, lymphopenia, and neutropenia, reflective of the lymphodepletion regimen they received. Most adverse events were consistent with those typically experienced by cancer patients undergoing cytotoxic chemotherapy or cancer immunotherapy. CRS of any grade had occurred in 4 out of 6 patients, none of which were grade 3 or higher. No DLTs had been reported at the time of data cutoff, and 1 SAE of CRS was reported in a patient in cohort 2 that was considered related to T cells. No other SAEs had been reported. This slide shows the tumor responses observed. As the data cutoff, there were 2 confirmed RECIST responses, one in a patient with head and neck cancer that lasted until around week 12 with a 63% reduction in target lesions. The other response was in a patient with EGJ cancer, which lasted beyond week 36 with a 51% reduction in target lesions. Importantly, although not all reaching a RECIST response, 5 out of 6 patients experienced initial reductions in tumor size. Taken together with the data we saw from the first generation phase 1 trial, we remain confident that MAGE-A4 is an attractive target across multiple tumor types, addressable with our highly active SPEAR T cells. These data continue to support enrollment in the continuing expansion phase of the Surpass trial and the planned phase 2 trial in gastroesophageal cancers based on the data from the 3 patients on the right with the green stars demonstrating tumor reductions. In conclusion, most adverse events were consistent with those typically experienced by cancer patients undergoing cytotoxic chemotherapy or cancer immunotherapy. The emerging waterfall plot is very encouraging and indicative of an active product with 2 RECIST responses and 5 out of 6 patients demonstrating initial tumor reductions. We're focused on enrolling patients with gastroesophageal, head and neck, lung, and bladder cancers, indications for which SPEAR T cells targeting MAGE-A4 have already shown promise. We're also planning to start a phase 2 trial with this next generation product in patients with gastroesophageal cancers that will initiate next year. I believe that our SPEAR T cells targeting MAGE-A4 represent a groundbreaking opportunity and have the potential to benefit many people with cancer. Lastly, I want to offer our appreciation for the investigators and their teams at our study centers, and especially the patients, their caregivers, and loved ones who participate in our trials, particularly in these challenging times. As a quick reminder, this is a modified 3+3 dose escalation trial. We have completed the first 3 cohorts and are currently enrolling in the expansion cohort. Most patients enrolled in this trial have advanced disease and have received multiple prior lines of therapy. In addition to patients with AFP expressing hepatocellular cancers, we have opened a cohort to treat patients with other AFP expressing tumors. We will share data about this cohort at a later date. We have treated 3 patients in cohort 3 and 1 in the expansion cohort as of the data cutoff. Of these 4 patients treated with doses of approximately 5 billion cells, 1 had a complete response, 1 had stable disease, and 2 progressed. The patient who had a complete response has now developed new lesions at week 32, with an overall duration of response of approximately 7 months. This patient also had a sustained reduction in serum AFP. In cohorts 1 and 2, we have treated 5 patients. In these 2 cohorts, although there were no responses according to RECIST criteria, we did see further evidence of anti-tumor activity at these lower doses, with all 5 patients having stable disease as best overall response. These data indicate that our SPEAR T cells targeting AFP are active and able to engage with the tumor, as shown in the patient who had a complete response, an achievement unto itself in this population. We are treating patients in the expansion cohort with doses up to 10 billion cells. We are also looking at translational data so that, if necessary, we can explore ways to make this an even more effective therapy,